So without further ado, let's start the topic. So we are very much interested. How can I change this? Just here, no?
It doesn't work, the green button.
If I go back.
Oh no.
Oh yeah, thanks. Okay. So we are very much interested in the gut-brain axis because anytime we have a perturbation in the intestine, which could be alteration in the gut microbiota, a condition we call dysbiosis, this is usually linked to inflammation in the gut. And as clinicians might know, many people affected by neurological disorders, neurological condition, they report to have gastrointestinal dysfunction. And people might also start to investigate if even multiple sclerosis could start in the gut before going to the brain. So we're very much interested in this crosstalk between the two organs. And another factor, and we are looking at the microbiota mainly, but another factor that we are studying here, it's how hard should I press? Okay, it's nutrition, because nutrition, it's actually a complementary and alternative and very efficient tool that we could use in the clinic also to treat neurological diseases. And dietary treatment have been used in different disorders like drug-refractory epilepsy, autism spectrum disorder, multiple sclerosis, and Alzheimer. But we don't know yet, we don't understand there yet the mechanism of function. And therefore, some of this diet, actually, they also cannot be kept lifelong, especially, for example, the ketogenic diet, which I'm talking about that today, because of the side effect. So we need to really understand how diet works, and if we can mimic this effect, one way we think it's through the microbiota, to have prebiotics or prebiotics treatment that can be much easily provided in a safer manner to a broad spectrum of patients. So we work with mice, and next slide, please. So we work with mice mainly, and we started a project looking at the ketogenic diet, which is a high-fat diet. 80 to 90% of the calories are coming from fat. It's very low in carbohydrates. It has been used in many different animal models for multiple sclerosis on the top, drug-refractory epilepsy in the middle, and Alzheimer's disease. And if the mice are treated with this diet, they actually perform better. So they have a lower clinical score of EAE. We need to use an higher threshold to induce seizures induction, and they perform most likely like the wild-type animals in Alzheimer's model. So we wanted to understand what is happening there. Next slide. Yeah. And we are, of course, not the only one that do that. So very recently, the group of Peter and Torbaum showed that actually the ketogenic diet effect is really mediated by the microbiota, because the ketone bodies which are produced in the gut by intestinal epithelial cells are taken up by gut microbiota, especially lactobacillus family member, which produce interlactate as a downstream effect, which blocks Th17 cell development. And in this work, they also check in people, and in untreated MS and in very severe MS, these bacteria are lower in abundance. So there's a hint that this might be also useful in the clinic, but animal models, they certainly show that the effect of diet is mediated by the microbiota. Another group showed that actually in a different models, in the seizure models of epilepsy, alteration of the gut microbiota with an increase in Akkermann's municipal and parabacteriitis leads to a differentiation in the brain of GABA and glutamate content. This was leading to protection for seizure. So we were interested to understand that, and we started in steady-state condition because I think we still don't understand yet what happens during homeostasis. So next slide. So what we did is to take a ketogenic diet and a controlled diet. Here I need to stress the fact that we use purified diet. Their composition is open, so we know exactly how they are made up. And the same also for the controlled diet. So these are different. The controlled diet is not the standard chow that every mouse facility uses, because that's undefined. So we want it to be in defined condition. And we always take SPF mice, which are specific pathogen-free animals, colonized with a very diverse and undefined microbiota, versus germ-free mice, because there we want to disentangle the effect of diet alone on the host and the microbiota-mediated effect. So our diet, as you can see, it's 90% calories from fat. Next slide. And so we started to look at the model. So we can induce ketosis. We measure beta-hydroxybutyrate in the serum. And this reaches very similar level in both SPF and germ-free conditions. So we have ketosis in these animals. The mice maintain their weight. They lose a bit of weight, especially if they wear SPF, and especially males. But this is quite standard whenever the mice are switched to a purified diet. And afterwards, they keep their weight. So we treat the mice for a month, and as you can see here, one reason is that their weight is stable, and also their microbiota gets stable. And this, we know it from many studies from Andrew McPherson group. You need a certain time to reach that. So we wanted to, in a stable condition, but we wanted to use adult animals. So the first experiment we did is to really look directly in the brain, to see if there's any effect of ketogenic diet and microbiota in the brain. We did a spatial transcriptomic analysis to also understand if different regions of the brain are affected differently. And what we observed, so we were able to distinguish different clusters, 12 different clusters, which are 12 different regions on the tissues. And as you can see here, despite very tiny regions like the choroid plexus, all the others are quite well conserved in all the tissues. So we could compare them, and we look at the data at the genes that are differentially regulated. So in the first line are genes that are upregulated in a significant manner in SPF condition in mice treated with the ketogenic diet versus the control diet. And as you can see, there are many, many different genes. As soon as we take out the microbiota, so the second line in the germ-free condition, we lose completely this effect. So this effect seems to be microbiota dependent. And as you can see, it's not all over the brain, but it's in specific area. And we selected the hippocampus, which is cluster 5 and 9 together, and it's the ones having the highest amount of genes differentially regulated, mainly upregulated, which was also very interesting. And this is a region involving memory and learning. There's also thalamus and hypothalamus, which are the region 0 and 4 together, very important in movement. So I will show data on this, which are maybe very relevant for this audience, and also the amygdala region. So we have an effect in specific area of the brains, which have a very different function, and we want to understand what's happened there. So from a very first string analysis, next slide, please. We show, one back. So we show, for example, we observe that in hypothalamus and thalamus together, we have upregulation of pathways involved in synapse formation, mitochondrial activity, which is very conserved, also in the amygdala in the next slide. But myelination is very specific for this area. So we have upregulation of genes MBP and MOBP on the left side, and PLP and MOBP also on the right side. So this is very interesting. We didn't go deep enough in this analysis yet, but this is a next project that we will look at that. So in the next slides will be the amygdala, and we show here also synapse formation. It's conserved, and in the region that we started to look at, which is the hippocampus in the next one, it was quite striking that also here we have mitochondrial activity. Ribosomal complex was very highly upregulated here, which could be a sign of neurogenesis. We don't know that yet. We will do additional analysis. Lipid transports, nervous system maturation, and maintenance were also upregulated. So when we did this experiment, we couldn't go yet to the single cell level. The technology was not developed there yet. Now now they have a kit to do that. So what we did, it's a single nuclei sequencing. The next slide, please. Starting to focus on the hippocampus, because I show you at the beginning was the region with the most differentially regulated genes there. And we look there, so with the UMAP, we are able to distinguish all the different cells that are there. We didn't pre-select for anything, so we really get everything out of this. And I highlighted the cells where we are focusing now at the moment, because it seems to be the cells which are mostly affected by the presence of ketogenic diet and the microbiota. So if we look at the neurons, so we have two subsets, the CA1 ProS neurons, which are the pyramidal neurons mainly. They are the ones that upregulate genes in the ribosome. So they seem to really have a high potential of transcription potential and probably peptide production. With this, we didn't address that, we will do it soon. They also upregulate hallmark of oxidative phosphorylation and downregulate pro-inflammatory response, which is also very interesting in our case. Another subset of neurons are the DG neurons, and they also tend to show the same phenotype, even though they have, in this case, they have a higher amount of genes sitting in the biological process of neurodevelopment and neurogenesis. So we need to do more analysis on this, but it seems that we could confirm what we observe in the spatial transcriptomic, which seems to have a suggestion that neurogenesis happens in these conditions. If we look at the astrocytes and microglia, so astrocytes, they are the ones interacting with the neurons, and as we observe also in the spatial transcriptomic, they seem to upregulate genes involved in synapse formation. This is also very important, probably very interesting and relevant for example, drug refractory epilepsy. They also have a role in cholesterol metabolism. And the same function in lipid metabolism was also observed in the microglia, which upregulates also genes involved in bile acid metabolism. This was quite interesting to observe, especially for the data I'm going to show you soon on metabolism. So we were happy to see there was actually a strong phenotype in the brain in steady-state conditions. So again, our mice are totally healthy. So we looked then back in the gut where we were already expecting to see difference. And in fact, we have a different microbial composition, which we can appreciate at the alpha and the beta diversity in the left and right side. So if we look more deeply, so these are SPF mice. If we look deeply at what is changing, and the next slide. So we have changes more in the upper intestine and the lower intestine, so in the small intestine and the colon. And there are changes which has been already reported in the literature with a decrease in abundance of actinobacteria, especially bifidobacteria, which is the last errors in this graph. I'm sorry that the colors are lost. But in the left sides are the ones associated with controlled diet, and the right sides are the ones associated with the ketogenic diet. There's also a tendency to have an increase in firmicutes abundance, especially in the small intestine, compared to the colon, and a different ratio in firmicutes and bacteroides, let's say. So I have highlighted some of the bacteria which seems to be really associated, and the one on the upper sides are with the ketogenic diet, and I highlighted some Clostridia family members, which we find very important because we can confirm the same with the different models. So why we use, which we use? In the SPF condition, we have this different microbiota composition, and we also did a meta-transcriptomic analysis there to understand what the bacteria produce. The data are still under investigation because the annotation in the mouse microbiome is still lacking a bit of deepness. So we took, in parallel, we took a different step. So we use a gnotobiotic mouse model we have in the lab. They have been generated by Hendrik McPherson and Barbara Stecker in Munich years ago. And we have this settings in many different facilities at the moment. So these are called the SDMDM, which is a very complicated names to just say that the mice have 12 different commensals, which are very stable, which are depicted there, very stable in vivo. we can culture all of them independently in vitro, and their genome is fully sequenced. So we can really do metatranscriptomic analysis in a very precise manner in vivo in this case, which has been already shown by Andrew McPherson group some years ago in these other studies where they use high-fat diet versus high-starch diet. So we wanted to use the same system also in our case, especially because, I don't go into details, but this microbiota, it has all the characteristics very similar to the SPF. It's not perfect, but it has bacteria which have been shown in the literature to change upon ketogenic diets. So in the next slide, we did a metatranscriptomic with this model, treating them with ketogenic diet, a controlled diet. And as you can see here in the first bar graphs, we highlighted in bold the bacteria which are differentially abundant. And they are Clostridia family members. So these Enterocloster Clostridioiformes and Flavonifractoprides are Clostridia which seems to be increasing in abundance. And if we do a metatranscriptomic analysis, we are really able to see which genes they upregulate or downregulate in a specific diet-dependent manners. And as you can see, there are bacteria like Akkermansia or Burkholderia bacteria tend to downregulate only genes, and others which upregulate genes like Lactobacillus reuteri, which is very important, has been shown. It's one of the bacteria that the group of Peter Thorbaum showed to be important in the ketogenic diet response. But we have also Clostridium member like Lactobacillus, Lactoclostridiums and Flavonifractoprides, which both downregulate and upregulate genes. And if we dig more into their metabolism, next slide please, we see that actually Clostridia tend to have upregulation of pathways specific for this species. They also upregulate pathways which compensate pathways that are downregulated in other species, like the Bacteroidetes, but there are specific pathways which are specific for this species. And if we look into the details of this is a preliminary analysis, but there seems to be pathways involving biosynthesis of amino acids and cofactors. This might be very important because we need to have amino acids migrating to the brain to produce neuroactive peptides, for example. So we are now understanding what each of these bacteria do, and we are in the process to do that. And what we did in parallel also to look at the metabolites profile all over our tissues, and we screened the brain, the serum, and different parts of the intestine in both conditions, germ-free and SPF, in the presence of control or ketogenic diet. And we wanted to understand and find out some characteristics which are specific for ketogenic diet and the microbiota, which are highlighted here. And I mean, you see also within that, we see some difference. This is due to the sex of the animal. So I'm not digging into that topic yet, but we have difference between male and female in our studies, and all the data that I show you here are done in males. So looking at the metabolites in the different tissues, we observe difference in the colon. Next slide. Especially we were surprised, but also it makes sense that we have an upregulation, an higher abundance of bile acids metabolites, so taurocholic acids, for example, in the colon of mice treated with the ketogenic diet, compared to the others, and the same was true also in the serum. So there are more bile acids in these animals, at least from an untargeted analysis, which we need to confirm further on. So in the brain, of course, we had a bit less difference, but we had, for example, we see ketone bodies, but we see also succinate, so from a TCA cycle, which is a downstream metabolite from the ketone bodies. So we think that something is happening. This is really mediated by the microbiota. And now what we are trying to see if with a targeted analysis of the bile acids, not only the primary, but also the secondary bile acids, we could rule out if they're really involved in the phenotype that we see. So we did a first targeted analysis of TCA and GCA. These are the primary bile acids. We are we have ongoing analysis on the secondary bile acids, which are very dependent on the microbiota. But already here we see a difference. So the TCA seems to be really higher in mice treated with a ketogenic diet, and especially in the presence of the microbiota. So we think that what is happening here is that the microbiota changes, and we might have an upregulation of metabolism, of bile acids metabolism and TCA cycles, which we can detect also in the periphery, in the serum. And this might be the mediators of the phenotype that we see in the brain, with an upregulation of myelination, oxidative phosphorylation, and neurogenesis, for example. So we are now want to perform more mechanistic experiments where we can supplement the bile acids, or we have monocolonization experiment with some Clostridia family members to rule out if this is really the cause of that, and we want to perform behavioral tests. So in the last few slides, I want to show you actually a project which started already in Bern, but was a bit unlucky because with moving to Berlin and establish a new animal facility there, it got a bit delayed. But with the help of Professor Weckele, actually, we have the animals, TCR1640 transgenic mice, the relapsing remitting models in both germ-free and SPF condition, where we wanted to understand there how the diet introduced at different time points in life could actually affect the development of EAE, the experimental autoimmune encephalomyelitis. And we are very much interested on the T cell side. So for this project, we choose to use Mediterranean diet and Western-like diet because it has been shown that the first one is usually linked to a more protective and anti-inflammatory phenotype, whereas the second one with a more pro-inflammatory phenotype. So what we did is actually to start with germ-free animals, which were colonized with a normal microbiota coming from SPF mice, so SPF black 6 mice, which are not susceptible to the disease, to the EAE, and we colonized germ-free mice. We bred these animals and we studied the offspring, which are treated with a different type of diet, so the Mediterranean, the Western, or a controlled diet, at different stages of life, and we started very early in life, so giving the diet already during the pregnancy, during the mating. And as we first saw, is actually the mice treated with the Western diet lost lots of weight, and we almost lost that group of colonies. So we had really, really few mice at the end of the experiments. And so we don't know yet what is happening there, but the phenotype was actually quite strong. And we're now going on in Berlin to investigate how, what's the phenotype of the T cells there and how the disease progressed. This was very specific for the relapsing remitting mice, as the black 6 didn't show any sign of the same phenotype. So, as I told you, we had a bit of issues to use these animals in different facilities, so we had to restart the colony. But what we did in the meantime, next slide, to treat T cells, which are differentiated in vitro towards the Th17 cells, with the intestinal content coming from the small intestine or the colon of germ-free or SPF mice treated with the Mediterranean, Western diet, or controlled diet. And we use the same protocol that Caroline Port and Jessica Ribedeau do in Lausanne, so thanks to them for sharing their settings. And as you can see, we might see some different phenotype if the cells are cultured with the small intestinal content, depending on the diet, especially looking at TNF-alpha positive cells or IL-17 positive cells. So we are now, we didn't test yet what is contained in the content of these animals, so we didn't perform any metabolomic analysis, but this will be the next step. So with that, I would like to thank my team. So Andrina Ruch, she's the PhD student who performed the first project, and Johan Kanson, the second project, is actually from Lucerne. And I need to thank really my mentors, Kathy McCoy and Anu McPherson, who still gave me the opportunity to start all this project in Bern, and Andrew for still giving me the possibility to have access to the Clean Mouse facility in Bern, which was a huge help for our research to not stop it completely. Our collaborators and all the core facility that supported us during our studies, and thank you for your attention. I'm happy to hear your opinion. Thank you.